In the thick of night, the tolling of church bells echoed mournfully through deserted streets, mingling with the cries of the dying. The year was 1347, and Europe was gripped by an unimaginable terror. Silent roads bore witness to wooden carts creaking over cobblestone, piled high with lifeless bodies. The very air seemed heavy with the breath of death. In just a few short years, the Black Death would claim the lives of over 50 million people, erasing nearly one-third of Europe's population and forever altering the course of human history. More than 7 centuries later, inside a cutting-edge laboratory at McMaster University in Canada, scientists carefully open a small container holding ancient teeth and bones, the silent remains of those who perished long ago. A single haunting question lingers in the air. Can ancient DNA reveal the hidden secrets of a plague that shaped the destiny of humankind? Join us on this journey of discovery. And don't forget to like, comment where you're watching from, and subscribe so you never miss the next chapter of our story. For centuries, historians and scientists believed they understood the cause of the Black Death. The accepted explanation was simple and seemingly undeniable. A single bacterium, Yersinia pestis, spread by fleas living on black rats, was solely responsible for the most devastating pandemic in human history. This idea was rooted in historical records and observations. Medieval chroniclers like Giovanni Boccaccio, author of the Decameron, described scenes of chaos and death, noting that swarms of rats seemed to appear before outbreaks erupted. In later centuries, epidemiologists relied on these descriptions, combined with modern observations of plague outbreaks in Asia and India, to strengthen the theory. Archaeological discoveries seem to support this view. In London, the East Smithfield burial site, unearthed during excavations for the Crossrail project, revealed mass graves where thousands of victims had been hastily interred. Similar plague pits were discovered across Europe, from Barcelona to Lübeck and Bergen, op Zoom, each pointing to a fast-moving, catastrophic disease. The scientific foundation of this belief was laid in the late 19th century when Alexandre Yersin, a Swiss-French bacteriologist working in Hong Kong, first identified Yersinia pestis in 1894. His groundbreaking discovery convinced generations of researchers that the Black Death was simply an ancient version of the same plague seen in modern times. Throughout the 20th century, organizations like the World Health Organization, WHO, reinforced this view, describing the pandemic as the result of rats, fleas, and poor sanitation in crowded medieval cities. It became the textbook explanation, repeated endlessly in classrooms and documentaries. For centuries, this narrative was so widely accepted that few dared to question it. It was a neat, logical story, one that seemed to explain the unimaginable tragedy. But beneath the surface, cracks were beginning to form, as new evidence hinted that the truth might be far more complex than anyone had imagined. By the dawn of the 21st century, whispers of doubt began to emerge. Some historians and scientists noticed troubling inconsistencies in the old narrative. In many plague-stricken cities, there was little archaeological evidence of rat populations large enough to explain the speed and scale of the Black Death's devastation. Even more puzzling, records suggested that the disease spread far too quickly for fleas and rats alone to be the primary carriers. Something didn't add up. The true turning point came with the rise of ancient DNA research. Between 2011 and 2015, a series of groundbreaking studies revolutionized our understanding of the pandemic. For the first time, scientists could extract and analyze genetic material from the very victims of the Black Death. At the forefront of this work were 2 pioneering teams, Johannes Krause and Kirsten Bose at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, Hendrik Poinar and his colleagues at the McMaster Ancient DNA Center in Canada. These researchers worked with samples from plague burial sites across Europe, including London, Barcelona, Lübeck, and Bergen, op Zoom. In sterile clean rooms, they carefully drilled into centuries-old teeth, extracting tiny fragments of degraded DNA. What they discovered would change history. When Krause's team sequenced the genetic code of Yersinia pestis from medieval victims, they were stunned. The ancient strain was genetically distinct from modern plague bacteria. It wasn't simply a relic of the same disease we know today. It was an earlier, more virulent form that had since evolved. Even more surprising, they found patterns of spread that couldn't be fully explained by rat-based transmission. The data hinted at human-to-human transmission, possibly through respiratory droplets or even lice and human fleas, which would explain the rapid movement of the disease across entire continents. Then, in 2022, a study published in Nature by the McMaster and Max Planck teams delivered another shockwave. They discovered that the Black Death didn't just kill, it reshaped the genetic landscape of Europe. A particular gene, ERAP2, linked to immune system function, appeared far more often in those who survived the plague. People carrying this gene variant were more likely to withstand the disease, meaning their descendants, many of us alive today, still carry its legacy. This revelation suggested that the Black Death was not only a historical catastrophe, but also a force of evolution, permanently altering the very DNA of humankind. The path to understanding the Black Death through ancient DNA was anything but simple. The teeth and bones of victims had been buried for more than 7 centuries, their genetic material decaying with each passing year. To the naked eye, these remains seemed like nothing more than brittle fragments of history. But to the scientists determined to uncover their secrets, they were priceless time capsules, silent witnesses to one of humanity's greatest tragedies. Inside the McMaster Ancient DNA Center in Canada and the Max Planck Institute for Evolutionary Anthropology in Germany, researchers worked under extraordinary conditions. The laboratories looked more like high-security clean rooms than the typical science facilities. Scientists wore full-body suits, masks, and double layers of gloves to prevent even a single modern cell, a stray hair, a flake of skin, from contaminating the fragile medieval DNA. Every surface gleamed under ultraviolet light, designed to destroy any trace of outside genetic material. The process began with the careful selection of samples. Not every bone or tooth could be used. Teeth, especially molars, were the gold standard because their inner pulp chamber often preserved DNA better than other skeletal parts. Teams traveled to archaeological sites like East Smithfield in London, where thousands of plague victims had been hastily buried during the peak of the pandemic. Each tooth was logged, photographed, and stored under strictly controlled conditions before being transported to the lab. Once inside the clean room, the teeth underwent a painstaking preparation process. Using sterile drills, scientists opened the teeth and ground the inner material into a fine powder. From this powder, they attempted to extract fragments of DNA, fragments so tiny and damaged that, in some cases, only a few molecules remained intact. This was the most delicate stage. Ancient DNA is incredibly fragile, often shorter than 50 base pairs, and riddled with chemical damage caused by centuries of exposure to moisture, bacteria, and soil. To put this in perspective, modern human DNA is usually hundreds of thousands of base pairs long, making the ancient material like shattered glass compared to a complete window. Next came the magic of modern technology. The extracted DNA was treated with specialized enzymes and chemical buffers to stabilize it. Then, using next-generation sequencing, NGS machines, scientists could read millions of tiny fragments simultaneously. Sophisticated computer algorithms assembled these fragments like pieces of a jigsaw puzzle, gradually revealing the complete genetic code of the bacteria, and, in some cases, the humans themselves. The first breakthrough came when the team successfully reconstructed the genome of the medieval Yersinia pestis strain. Under the leadership of Johannes Krause and Kirsten Boss, the Max Planck researchers compared this ancient genome to modern strains of plague bacteria. What they found was startling. The medieval strain wasn't merely an older version of today's plague, it was ancestral, a kind of patient zero for centuries of evolution. This suggested that the Black Death was caused by a form of plague far deadlier than the ones we encounter today. But the discoveries didn't stop there. The sequencing of human DNA from the same remains revealed something even more profound. Hendrik Poinar's team at McMaster noticed unusual patterns in genes related to the immune system. One gene, ERAP2, stood out. Individuals who carried a particular variant of this gene were far more common among those who had survived the Black Death. In other words, these people's bodies were better equipped to fight off the plague, giving them a survival advantage during the pandemic. This was an unprecedented insight into how a disease could directly shape human evolution. The findings, later published in Nature, showed that the Black Death acted like a brutal filter, removing individuals without the protective ERAP2 variant from the gene pool. Over time, the surviving population passed this advantageous gene on to their descendants. Astonishingly, this genetic legacy persists today, visible in the DNA of millions of modern Europeans and their descendants around the world. However, there was a dark twist. While the ERAP2 variant once helped save lives, it now comes with a cost. Modern medical studies have linked the same gene to a higher risk of autoimmune disorders, including Crohn's disease and rheumatoid arthritis. This means that a genetic adaptation born out of desperation during the Black Death continues to affect human health nearly 7 centuries later. The implications of this discovery were staggering. It suggested that pandemics are not merely historical events, but active forces of evolution, shaping the destiny of our species in real time. Just as the Black Death left its mark on the DNA of medieval Europeans, modern pandemics like COVID-19 may be subtly influencing our genetic future. The research also shed light on another long-standing mystery: how the plague spread so rapidly across medieval Europe. Analysis of DNA from human parasites such as lice, combined with historical records, suggested that human-to-human transmission, perhaps through droplets or direct contact, played a much larger role than previously believed. This explained why the disease could devastate entire cities within days, moving faster than rats or fleas ever could. To fully appreciate these findings, imagine standing in the East Smithfield burial site today. Beneath the calm green grass lie the remains of thousands who once lived and died in terror. Through advanced technology, scientists have given these silent voices a chance to speak again, not through words, but through the intricate code written in their DNA. It is a reminder that the past is never truly gone. It lives within us, influencing who we are and who we may yet become. The culmination of years of meticulous work came in October 2022, when the joint research teams from McMaster University and the Max Planck Institute for Evolutionary Anthropology published their landmark study in the journal Nature. The results sent shockwaves through the scientific community and beyond. For centuries, the Black Death had been remembered solely as a historic tragedy, a dark chapter marked by unimaginable suffering and loss. But now, evidence showed it was something even more profound, a direct agent of evolution, an invisible force that had sculpted the very fabric of human DNA. At the center of this revelation was the ERAP2 gene, a key player in the body's immune defense system. Through painstaking analysis of teeth and bone samples from plague victims and survivors, the researchers uncovered a stark pattern. Individuals who carried a specific variant of the ERAP2 gene were far more likely to survive the Black Death. Those without it perished in staggering numbers. This finding was more than just a piece of historical trivia. It was a clear example of natural selection in action. The Black Death had acted as a brutal filter, wiping out huge portions of the population, while allowing those with genetic resistance to pass their DNA on to future generations. In just a few short years, the genetic makeup of entire communities had shifted. But there was a haunting twist. While the ERAP2 variant had once been a life-saving adaptation, modern research revealed a dark legacy. Today, people carrying the same variant are at a higher risk for autoimmune disorders, including Crohn's disease and rheumatoid arthritis. In other words, the very gene that protected our ancestors centuries ago now contributes to chronic illnesses in millions of people alive today. The implications were staggering. This wasn't just history. It was a story written inside our bodies. The Black Death had never truly ended. Its echoes lived on in every cell, a genetic scar passed silently through the generations. As the study's findings spread across the world, a profound realization took hold. Pandemics don't just change societies. They change humanity itself, leaving marks that last far beyond memory or recorded history. For the first time, scientists could see in real time how a single event had forever altered who we are. The revelations about the Black Death reach far beyond medieval history. They force us to reconsider what it means to be human, to see our bodies not just as vessels of the present, but as living records of the past. Within every cell of our DNA lies the story of survival, of ancestors who faced unimaginable horrors and endured. The discovery of the ERAP2 gene's role highlights how pandemics are not merely fleeting events. They are powerful evolutionary forces that can reshape entire populations. During the Black Death, this happened in a matter of decades, an evolutionary blink of an eye. For those who lived through it, survival was a matter of chance and biology, not choice. Some families were wiped out completely, while others endured because of a single genetic variation, a twist of fate encoded in their DNA. When we compare this to our modern world, the parallels are striking. During the COVID-19 pandemic, scientists observed how genetic factors influenced why some people experienced severe illness, while others had only mild symptoms, or none at all. Just as medieval Europeans lived and died by the hidden instructions of their genes, so too do we, though we rarely think about it. This research also brings a sobering perspective on health today. The same ERAP2 variant that saved countless lives in the 14th century now contributes to autoimmune disorders like Crohn's disease and rheumatoid arthritis. It is a profound reminder that evolution has no foresight. A genetic advantage in one era can become a liability in another. On a deeper, human level, these findings connect us directly to the past. The people who buried their loved ones in mass graves, who rang the church bells in despair, were not distant strangers. Their DNA flows through our veins. Their choices, their struggles, and their survival shape the very blueprint of who we are today. By studying ancient DNA, we do more than solve historical mysteries. We illuminate the intricate web that binds past and present. It shows us that history is never truly gone. It lives within us, a silent, enduring legacy guiding humanity toward its future. As the camera drifts over a quiet green field in modern-day London, it is hard to imagine the horrors that once unfolded beneath the soil. This peaceful meadow was once the East Smithfield burial ground, where thousands of victims of the Black Death were laid to rest in haste and despair. Today their voices are silent, yet through the science of ancient DNA, they speak to us once more. The research led by Johannes Krause, Hendrik Poinar, and their teams has revealed that the Black Death was more than a medieval tragedy. It was a turning point in human evolution, leaving marks still visible in our very cells. Every heartbeat we feel today carries the legacy of those who lived and died nearly 7 centuries ago. Their story is not only one of suffering, but of resilience. It reminds us that humanity has faced unimaginable challenges before, and survived. In the face of modern pandemics and future unknowns, their genetic whispers guide us forward. If this journey through time has moved you, please like this video, share it with others, and subscribe to our channel. Together, let us continue uncovering the hidden stories written in the code of life itself.